So in this video we'll be looking at how non-steroid hormones have their effects in cells, and also what are some of the bigger picture controls of endocrine activity, so the activity of the endocrine glands. So we will begin with talking about non-steroid hormones. So non-steroid hormones, they include, some example hormones include epinephrine and norepinephrine, the two that are coming from the adrenal medulla. The thyroid hormones, the two hormones that the thyroid gland produces that have their function around metabolic rates. Growth hormone, oxytocin, the cuddle hormone. And most of these work by a process referred to as signal transduction. Signal transduction here. Signal transduction means that binding, if this is the cell, and we have a receptor here that has a surface both inside and outside the cell, when the hormone binds here at what's referred to as the binding site, which is on that receptor protein outside the cell, it causes a change here at the activity site that will result in, eventually, through a series of changes, that will result in a change inside the cell. So with signal transduction, we're taking a signal out here in one form, the hormone, and transducing it, there's the weird word, taking it across the cell membrane and converting it into a different type of signal on the inside of the cell. And then as a result of signal transduction, we see all the changes that are going on inside the cell that we associate with that particular hormone. So we're going to use, once again, the drawing from the book to try to make some sense of this. The drawing is found on page 495. And this is how signal transduction looks inside of a cell. So the cell membrane is going to be studded with these receptor proteins. Now they're only showing 3. Here's one here, here's one here, and here's one here. But they would be all around the cell, in the cell membrane. And every one of these receptors has, outside the cell, a binding site, a spot that is attractive to the hormone molecule. And on the inside of it, what's called an activity site. And the activity site will change whenever the binding site gets filled with a protein. The way that I think of these changes as happening is that when this hormone molecule comes and binds right here, so that spot is very attractive to the hormone molecule, and so if the hormone molecules are found in the extracellular fluid because they've been dumped there out of the bloodstream, then the electrons that are swirling around the atoms in the hormone molecule, they are going to cause the electrons that are there in this protein receptor to readjust. So anytime you bring two electrons together, they're going to be repelled by each other. And so that repellent behavior causes some electrons up here to shift away from the non-steroid hormone where it bound. And that then causes a cascade of shifts all the way through the protein molecule. And it changes the shape here of the activity site, because the electrons are going to be moving around and adjusting because of what's happening out here outside the cell. Associated with this receptor molecule, this protein molecule or glycoprotein molecule that receives the hormone, is something called a G protein. So this here is the G protein. Now, it's another protein, also made up of atoms that have electrons around it. So as soon as we adjust these electrons here at the activity site, well, all of its electrons are going to readjust too, and it will take on a slightly different shape. And I think of that as activating the G protein. So, G protein is, well, the receptor molecule gets activated. That activates here at the activity site the changes, that activation, that shape change, the, however you want to think of it, the electrons that have jumped around a little bit, they are going to activate the G protein. Now these arrows that doesn't mean that the G protein converts into something else. These arrows are just the flow of action. So when the G protein gets activated, very closely associated proteins, this is adenylate cyclase, will also be activated. Adenylate cyclase has that ASE ending, so we know it's an enzyme. Adenylate cyclase is an enzyme that will take ATP, right there, and they're showing it, like a big splash, because it's the energetic molecule, if you remember that. So we've got this energetic ATP molecule, and it takes it and it strips away two of the phosphate groups that are attached to it, leaving us with AMP. Remember, ATP stands for adenosine triphosphate. So when adenylate cyclase gets activated, adenylate because of the adenosine that's found in ATP, when adenylate cyclase gets activated, it will take any ATPs that come near it and convert them into adenosine, from adenosine triphosphate, strip away two phosphate groups, and what we're left with then is adenosine monophosphate. And because of some other activity that this enzyme does to the ATP molecule, it turns it into a cyclic version of it, of AMP. And so CAMP, that stands for, sorry about that once again. CAMP stands for cyclic adenosine, and because now there's only one phosphate molecule left, monophosphate. So cyclic adenosine monophosphate. All right, so let's just review what we've seen so far. The nonsteroid hormone will bind the receptor, forming this version of the hormone receptor complex. The receptor will change its activity site. It becomes activated, which gets the G protein activated, which in turn activates adenylate cyclase, which will then do a chemical reaction in which ATP turns into, or is converted into, cyclic AMP. All right, so far so good? So if we think about this hormone as being the first messenger, so it's the messenger sent out by the body to change the activity of the cell, this molecule, cyclic adenosine monophosphate, that is our second messenger. So this is how hormone molecules that cannot enter the cell do their work. That makes me think I did not point out that little piece, which is that because most of these non-steroid hormone molecules are polar molecules, they cannot, like the steroid molecules, they cannot just slip right on through the cell membrane. They are they are something that the membrane is impermeable to. So if the membrane is semi-permeable, it's permeable to steroid hormones, it's impermeable to most non-steroid hormones. And so the cyclic AMP is going to take that function of the hormone molecule, because it's the thing that we transduce the signal into. So in the idea of signal transduction, Here, the first messenger is our signal, and we are transducing our signal into cyclic AMP, the second messenger. So hopefully that all now makes sense. Second messengers are chemicals that are built inside of the cell in response to a hormone binding outside the cell. I also kind of use this analogy to think of it. If once again the cell is the house and a non-steroid hormone can't just walk in the door of the house, this is like the mailman. It's got a package. Mailman has a package, but the mailman doesn't have a key to your house, so he can't just put the package inside. He instead has to ring the doorbell. And the ringing of the doorbell gets the yippy-yappy dog that you have inside the house, the little ankle biter, gets all excited about that. Woohoo! And it gets all excited. Cyclic AMP is our yippy-yappy dog that gets all excited and runs through the house trying to find anybody he can to say, hey, we need some activity here inside the cell because somebody's at the door and they might have something good for us. Yip, 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 yip. Okay, so hopefully that kind of helps you think about how cyclic AMP and second messengers like cyclic AMP do their work. Okay, so now we have a bunch of cyclic AMP running around inside the cell, barking at everything. Here's what it barks at. Cyclic AMP can interact with a type of enzyme called a protein kinase. Protein kinases, anything that is kinetic, creates its movement. So this kin here is going to be activity. We got activity going on. And so ASE, there's our enzyme ending. Protein kinases are proteins that will do something to activate another molecule. So CAMP will activate a protein kinase. Protein kinases, when they're active, will then finally, it's taken a long time to do it, but it will find a substrate, some sort of molecule inside the cell. Anytime you talk about enzyme reactions, a substrate is what the enzyme binds and helps cause the chemical reaction to occur around. So our inactive substrate becomes active. And as a response, we see cellular changes. The changes in the cell that we associate with the hormone. So, let's take a look at this one more time and we'll just go through the process. A non-steroid hormone, because it can't slip on through the cell membrane, will instead bind at a receptor found in the cell membrane. The receptor becomes active, or the activity site changes. That activates a G protein. The G protein then gets adenylate cyclase, a nearby enzyme, excited, and allows it to convert ATP into the second messenger, cyclic AMP. With cyclic AMP running around, protein kinases become activated. With active protein kinases running around, then molecules, the substrate molecules, will become active and will produce the changes that we associate with the hormone inside of the cell. We finally got there. Okay, so that is how a non-steroid hormone does its thing. So now I got some words for you. Here we have signal transduction, a receptor protein outside the cell, on the outside surface of the cell, has a binding site and an activity site on the inside of the cell. When the hormone attaches at the binding site, we see a change in the shape of the activity site. This is how the activity site becomes activated. Then we see, through that series of reactions that we discussed with G protein, adenylate cyclase, getting activated, we see the production of a second messenger. Once the second messenger is activated inside the cell, then it will turn on a protein kinase. That's how it works with CAMP. Other second messengers may have other functions or other ways of activating the cell. And cyclic adenosine monophosphate is one of our common second messengers. It's not the only second messenger. So again, not every second messenger is going to do it through in the same way with protein kinases, but that's a real common way, and this book has chosen to use that as the example mechanism. So here are the steps for making CAMP. The G protein gets activated, which then activates the enzyme adenylate cyclase, which is just another membrane protein found on the inside of the cell membrane. Adenylate cyclase converts ATP into the second messenger CAMP. Then our second messenger activates a bunch of protein kinases. They work because they can transfer phosphate groups from an ATP to the protein, and that then activates a protein that will cause some changes inside the cell. So, when we think about non-steroid hormones, what kind of responses do they produce in their target cells? And remember, every cell in the body is probably going to encounter this non-steroid hormone, but only if there is a receptor for the hormone will the cell be a target cell. So some of the responses include that we can make the membrane of the cell either permeable or impermeable to a specific item. We may activate enzymes inside the cell. Oops, wow. That will be responsible for the activity. Sorry about that. I know that's disorienting for everybody, including me. It may cause the synthesis of particular important proteins inside the cell. It may stimulate or inhibit a specific metabolic process. It can cause other hormones or different kinds of substances to be secreted. And in the case of either a ciliated cell or a sperm, we can see movements of the cell in response to a non-steroid hormone binding there at the cell membrane. So lots of different ways that a hormone, a non-steroid hormone, might affect its target cell. And so it doesn't mean that every steroid hormone is going to do every one of these effects. This is just the, sort of the potpourri of choices. How's that? A potpourri of choices that the non-steroid hormone has available to it. Okay, so what are some other second messengers that we may find inside cells? Well, lots of cells use lots of non-steroid hormones use CAMP, including, this list is not exhaustive, but thyroid stimulating hormone, adrenocorticotropic hormone, follicle stimulating hormone, luteinizing hormone, antidiuretic hormone, parathyroid hormone, epinephrine, norepinephrine, calcitonin, and even glucagon. So lots of different hormones use CAMP as a second messenger. And that's why that's the one that we focus on in this chapter, because it is probably the most common second messenger. But other second messengers inside the cell might be diacylglycerol. Calcium ions act as second messengers. Ooh, calcium ions. Remember when we talked about an action potential that is going down the tubules that infiltrate the muscle cells? Remember, calcium ions are being warded up in the endoplasmic reticulum, or I guess in that case the sarcoplasmic reticula, there in the in those little collecting areas that are found near the edges of a sarcomere. And in response to calcium ions being released, then we finally see contraction of the muscle. So calcium ions have similar effects in other cells where they will bind to things and cause changes in the activity of the cell. They frequently use calmodulin, which is something that we talked about when we talked about smooth muscle cells and how calcium ions interact with calmodulin rather than troponin. And they may also, inositol triphosphate may also cause calcium ions to be released from internal storage, allowing them to be a second messenger. And then there is another nucleotide, guanine, can also be used to build a second messenger. That one's called cyclic guanosine monophosphate. So guanine and adenine are both nucleotides used to build nucleic acids, and adenine is the base molecule, of adenosine triphosphate. Guanine can be used to build a similar molecule with a single phosphate group attached to it, and it can be produced in cyclic form, so that's cyclic guanosine monophosphate. Okay, so that's just some of the idea of the different kinds of second messengers that may be used in cells. Okay, so we've talked about steroid hormones and non-steroid hormones. The book also mentions a little bit about how prostaglandins work. Remember, these are paracrine secretions. They're secreted by a cell, but because they are so quickly inactivated, they never make it into circulation. Instead, they only affect nearby cells. And they're produced around the body. We have already talked about a lot of the prostaglandin secretions of the digestive system. And so some of the effects of these prostaglandins when they come in and because they are built from a fatty acid, arachidonic acid, remember? So because they are built from a fatty acid, they can make it through a cell membrane, and their effects include relaxing smooth muscle that we find in the airways, in the bronchioles, and in blood vessels. They're in the middle layers of those two tubes of the body. They also can cause contractions of the uterus there, as the smooth muscle is affected by prostaglandins. They stimulate secretion of hormones. They can promote inflammation. Lots of different effects depending on the prostaglandins. So we're not going to spend a lot of time focused on prostaglandins, but that's that little bit that the book does talk about them. So now we're stepping back to look again at the whole endocrine system. And so this is not just about non-steroid hormones. This is about how steroid and non-steroid hormones work. So secretion by endocrine glands is routinely controlled by negative feedback mechanisms. And just to remind you, with negative feedback, there's a signal that causes a hormone to be released. So if we think about blood sugar, there's our easy one to go back to because we've talked about it a lot. So here's blood sugar, and this is time. Sugar and time here. So you wake up, blood sugar's low, so you're hungry, you eat some breakfast, blood sugar rises. There's our signal that causes a hormone to be released. Here, as blood sugar gets out of homeostasis, we're going to see insulin be released. That causes the signal that caused insulin to be released to be decreased now. So we reduce the effect of the signal, and then the effect lessens, and eventually we will get low again, and if we don't eat, then we're going to see glucagon. Here's a signal then, low blood sugar signals for glucagon, and as a result, we go to the liver and we unpack the storage, glycogen, and blood sugar goes back up again. So negative feedback always reverses the direction of the controlled thing, whether it goes up or down. So negative feedback is the means by which hormonal control is managed. But wait, how do we get these hormones that are circulating around the blood, how do we get them out of the body so that they can stop having their effect? Well, just like we talk about nuclear bombs and nuclear material having a half-life, the half-life of a hormone is the amount of time that it takes for, because of whatever inactivation means is occurring, the half-life is the amount of time that it takes for the concentration in the blood plasma to go down by 50%. So half-life is because we get to the point where there is half the amount in the blood plasma. So hormones that have a short half-life then are ones that are shut down very quickly. Epinephrine and norepinephrine have a short half-life. So if you're driving down the highway and somebody tries to pull into your lane exactly where you are, you get all excited about that. You get that big shot of epinephrine and norepinephrine. In response, you get kind of all shaky because all your cells are busy making huge amounts of ATP, and you get all kind of jittery and shaky. You get that weird, nasty, metallic taste inside your mouth. All of that goes away relatively quickly within usually 1 or 2 minutes even. So relatively quick decrease of the response. That's because we can clear epinephrine from the blood plasma very, very rapidly. It has a short half-life. How do we get rid of it? Well, either because we are peeing it away at the kidney, or because it's broken down by enzymes in the liver, or maybe even enzymes that are out there in the bloodstream too. Hormones that have a longer half-life often are the ones that have longer lasting effects. So thyroid hormones and many of the steroid hormones are very slow to be removed from the blood, and that allows them to have effects for days following secretion because they're still out there floating around, not being removed from the blood plasma. Okay, last little concept here in this chapter, which is what are the specific control mechanisms used to determine whether or not a hormone will be released? Well, the first is that a hormone may be controlled by the release of another hormone. So, for instance, the hypothalamus, it secretes what are known as tropic hormones. Tropic hormones, these are regulatory hormones that cause other glands to produce the hormone. Okay, so I'm going to take you back to GnRH, which we talked about in the reproductive system. This is gonadotropin releasing hormone. So gonadotropin, it's a tropin because it is telling the gonads to release their hormones. So tropic, and you'll see that tropin word in the name of the hormone frequently, that causes the release. That is a reference there to the fact that it stimulates or inhibits the release of hormones. It changes the activity. So yeah, I've been saying that it's only about releasing the hormone. It could either stimulate the release of a hormone or inhibit the release of the hormone. It's going to cause activity. That's what tropic means. Oops, sorry about that. So, hormone secretion may be directed by other hormones, and the hypothalamus produces a whole lot of these kinds of regulatory hormones that act primarily at the pituitary gland. We'll talk about that in the next video. Hormone secretion may also be directed because of neural stimulation. This is why you can produce epinephrine and norepinephrine so dang quickly. You almost get, hit by that car that's changing lanes into your lane where you are. We get that huge shot of epinephrine because there are nerves from the sympathetic division of the autonomic nervous system that will cause, I say suprarenal glands, that's an old school term, the adrenal glands, or a different school, not old school. The adrenal glands and the suprarenal glands, these are the exact same glands. So adrenal glands release it very quickly because there's a neuron or nerves that run from the brain to the medulla of the adrenal glands and causes epinephrine to be released. So nervous stimulation can cause hormones to be released. And lastly, the changes in the internal environment of the body can also cause the release of hormones. So here's a great example where blood glucose is involved with the release of two different hormones of insulin and glucagon. Calcium levels will affect the release of calcitonin and parathyroid hormones, and so on. So internal environment may also be involved in controlling whether a hormone gets released. And as always, negative feedback is involved here. Once the calcium levels are back in homeostasis, then parathyroid hormone and calcitonin stop being released. And all of these are negative feedback. Once the brain stops telling the adrenal medulla that, somebody's trying to kill us, then epinephrine stops being released. Once testosterone and estrogen are being produced in the body, then GnRH will reduce the amount of secretion that occurs. Or the hypothalamus will reduce the amount of secretion of GnRH that's occurring. Alrighty, there we are. It has been a pleasure talking to you. Let me just double check that there's nothing hanging out there. Okay, yep, we're good. So that's everything for this video. I'll see you in the next one.